all night. So we're really happy and excited. Is this coming through? We're really happy and excited to have Hongkui Zeng here today. She is the Executive Vice President and Director of the Allen Institute for Brain Sciences. I think many of you already know her. And I had the pleasure of reviewing her CV in preparation for doing this. And her undergraduate was at the Wuhan University, a place that's become somewhat famous for the wrong reasons, I think. And then she did a PhD in molecular and cell biology at Brandeis University, and then a postdoc for 5 years at MIT. She actually spent a few years in industry, it looks like, a few different companies. And then one company, but did it change names? Yeah, it underwent some metamorphosis, but anyway, yeah. But then decided that she didn't like working for companies that kept changing their name, and went back into academia in 2006, going to the Allen Brain Institute. And she told me that it was very new then, it was just a few years old. And she's one of the few, maybe less than 5 faculty who are still there from the original batch of people that came there. So one of the survivors. And she's just been incredibly productive. And so, I mean, the funding is amazing. She's got 3 or 4 different currently funded UO1 and R01 grants, as well as some kind of a budget that comes from the Allen Institute itself. Lots of honors, but I'll just give you the highlights, which is the National Academy of Sciences and the National Academy of Medicine. So those are non-trivial kinds of distinctions to have. The publication record is really just staggering. It was 35 pages. I love trees, and so I didn't print all 35 pages, but, you know, Science, Nature, Cell, and, you know, repeat, basically. So all kinds of amazing productivity there. And then lots of trainees and lots of other involvement, sitting on study sections and all of the normal activities. So really just superlative background and accomplishments. And I don't want to take away your time to tell us about the most recent accomplishments, so I'm going to hand it over. Okay. Yeah.
Thank you very much, Abe, for the nice introduction. And I especially want to thank the graduate students here for the invitation to give a seminar here today. Okay. So the goal of neuroscience is to understand how brain works, how the hardware and software of the brain contributes to the generation of behavior, emotion, cognition, and things like that. This is a daunting task given the extreme complexity of the brain, with millions to billions of cells shown here for all kinds of animal species that we study, the brains of those animal species. And the thousands-fold more of connections among those neurons. To tackle this complexity, we think that it is essential to take a multi-level approach from cell type to circuit to in vivo physiology and behavior, and eventually perhaps to a computational understanding of how brain computes information and generates behavior. And to extract organizing principles of the brain at all these different levels. So at the Allen Institute for Brain Science, our primary focus is actually just at the first 2 levels, trying to understand the diversity, extraordinary diversity of cell types in the brain and how they get together and form functionally specific circuits. We established large-scale technology platforms and pipelines, generating comprehensive data sets as foundational resources to the community. And at the same time, we also try to use a data-driven approach to extract the principles and rules from the data as well. Okay, I know that there are a lot of bioinformatics students here, so I think that data-driven discovery is what we do, and it's really, really essential for us. So here summarizes the major approaches we take for studying individual cells and connectivity in the mammalian brain. Our front-end approach is the highly scalable single-cell genomics technologies, including RNA-seq, ATAC-seq, and the combination of the two, and spatially resolved transcriptomics. We use those platforms to profile hundreds of thousands to millions of cells and computationally cluster them into cell types to generate taxonomies and atlases for the brain. We also create genetic tools to target those specific cell types. Traditionally, we use individual marker genes, generating driver lines, reporter lines to target the different cell types. In recent years, we have switched into identifying putative enhancer elements that are specific to cell types by the combination of the single cell RNA-seq and single nucleus attack-seq data. And we test those enhancer elements in AAV viral vectors. And with a pretty good successful rate, we are able to establish enhancer viruses that target different types of cells in the brain. At the anatomical level, we use large-scale light microscopy and electron microscopy and to reconstruct the morphologies, full morphologies, and long-range projection patterns for individual neurons in light microscopy and synaptic level connections of densely reconstructed neurons within a local circuit in the electron microscopy data. To bridge molecular information and anatomical information, we use a set of multi-model approaches, such as triple modality patch-seq, and also barcoded connectomics, to be able to link molecular identities with the morphological, connectional, and physiological information of the cells. And in recent years, and in fact, we just started this year, we've also began to establish technology platforms for proteomics, trying to bridge the gap between genes, genetics, and structure and function by studying where what proteins are expressed in specific cell types and where they are located, for example, in nucleus or axons or synapses and things like that. So underlying all the experimental technology platforms, we also build strong computational capabilities that will allow us to manage the large-scale data we generated, enabling analysis and especially integrated analysis across different modalities, and finally dissemination of all the data, tools, and knowledge that we have generated to the community. And our ultimate goal is to bridge cell types and brain function by building a brain-wide cell type map across 4 dimensions. First, across species, from mouse to human and non-human primate. That's our primary focus. Across modalities, being able to integrate the different cellular properties together, from transcriptomics to protein to anatomy to physiology and eventually to function and disease. And we find that it's actually very efficient to do this kind of integration using single cell transcriptomics information. And integration across space. We don't want to just study one individual brain region. We want to study all the brain regions, all the different regions in the brain, and maybe in the body as well in the future. Understand the relationships of the cell types across different brain regions, and of course, how they are connected to form circuits that carry out behavior. And finally, across time, from development to aging, we want to understand how cell types change, how their properties change with time, and also under different functional and diseased conditions. So with this kind of very comprehensive and ambitious goals, we have many different projects happening at the Institute. For today's talk, I will be just focusing on telling you about the 3 main projects that we recently completed, and very much focusing on the initial stage of this big picture. And those would be focusing on mouse and using single cell genomics approaches to create a whole brain atlas for the mouse brain, and also then see how the cell types in the mouse brain change during development and during aging. Okay, so our starting point of generating cell type atlases is the single brain regions, in particular single cortical regions. And here I want to use this one study to highlight what we have done. And this is a part of the Brain Initiative Cell Census Network, which we are part of, in which dozens of labs work together, including those at San Diego, to use the multimodal approaches I told you about in the previous slide to profile cells from the primary motor cortex of 4 mammalian species: mouse, macaque monkey, marmoset, and the human. And we integrated all the data together, creating this taxonomy that's anchored on the mouse consensus transcriptomic taxonomy. So in addition to generating this atlas, we also learned a few principles about cell type organization, and I will summarize here. First of all, cell types are organized in a hierarchical manner, as shown in this taxonomy tree, with different levels of granularity, defining cell types from class to subclass to type or cluster. And then we are able to integrate the different modalities of data together, including transcriptomics, epigenomics, spatial distribution, morphology projection, and physiology. We find that there is the coexistence of discrete and continuous variations in all kinds of dimensions in this integrated space, with major discrete distinctions between cell types at a major branch level, the class and subclass level, whereas with more continuous variations of cellular properties at the terminal leaf or type or cluster level. We also observe the coexistence of cross-species conservation and divergence. Again, with the conservation mainly at the major branch level, class and subclass that we defined are absolutely conserved across all the 4 species we studied. But then at the cluster level, there's a lot of variations. There's never a single one-to-one correspondence for the different clusters. It's often several to several or multiple to multiple. And there are differential gene expression patterns of many genes, like, maybe like about 20% of the genes are between different species. So overall, this rules together really paints a complex landscape of cell types, even just from one brain region. But it's good to see that all this information can be summarized, in this kind of hierarchical manner. Okay, so then moving on, that's from one cortical region. Then this next study, we talk.
I'm just going to close out of the.
Oh, okay. But there are transcripts, live transcripts happening. Yeah, I hate that. Okay. So in this next study, we ask a question, do cell types vary between different cortical regions? Okay, so in this study, in which we only profile 2 different cortical regions, primary visual cortex at the back, anterolateral motor cortex in the front, we find an interesting dichotomy. We generated a taxonomy again, very similar to one cortical region, but we also find an interesting dichotomy between the 2 major classes of neurons. And in which the GABAergic inhibitory interneuron types are all common or shared between those 2 different cortical areas, whereas the glutamate excitatory neuron types are highly distinct between those 2 different cortical areas. And this dichotomy, we think, is due to a developmental, is related to the differential developmental origins of those 2 major classes of neurons, with the excitatory neurons generated within the cortical plate, which I will show you later, whereas the inhibitory neurons coming from subcortical ganglionic eminence areas and migrate into the cortex during development. So this dichotomy suggests that there's a gradient, spatial gradient within the cortical plate that's inherited in the transcriptomes of the excitatory neurons, but not in GABAergic inhibitory neurons. And in later studies, which I'm not going to show here, we find that when we profiled all the cortical regions, across the entire cortical sheet, we find many different kinds of spatial gradients across different cortical areas, horizontally medial lateral, anterior posterior, all these different dimensions, even though the cell types themselves are very similar or related between different cortical areas. And this yet another study in which we used a patch-seq as a way to profile the transcriptomic morphoelectrical information of the cells, and especially the GABAergic inhibitory neurons, which is known to have a tremendous diversity. from the mouse visual cortex, we're able to integrate the triple modality information together to define what we call the MET met types. And we defined the 28 met types as shown here that cover 5 major subclasses of GABAergic interneurons that are well known in the field. So the met types have intermediate granularity compared to the T-type or transcriptomics types, which we defined as 61 in mouse visual cortex. But they have higher cross-modality correspondence and also mutual predictability. The met types also recapitulate traditionally defined interneuron types as shown by the names assigned to the different met types. Those are the traditional names. And they further subdivide the traditionally known GABAergic types based on their layer specificity. For example, the somatostatins met types, a beautiful example showing 13 different met types that recapitulate all the known diversity of the somatostatin neurons, but then the met types are nicely organized across all 6 layers, from deep to superficial, not only in terms of their soma location, but also in their local axon projection patterns. And finally, the patch-seq data then the met types can serve as a Rosetta Stone to bridge molecular information and morphoelectrical information in completely disparate data sets. For example, recently we published a series of papers of the electron microscopy reconstruction of the mouse visual cortex. And in those studies, we used the patch-seq cell types to assign, and through a morphology match, to assign molecular identities to the neurons that are reconstructed from the electron microscopy data. And so linking molecular identities with their synaptic connectivity motif information. Okay, so with that, we're just further moving on, jump a few years ahead. This is yet another big collaboration within the BRAIN Initiative, in which then we completed the creation of a cell type atlas across the entire mouse brain. So we jumped from individual cortical regions to now the entire mouse brain. And from Alan's side, this showed what we've done, contributing to the whole mouse brain cell type atlas. We generated 2 large-scale data sets, a single-cell RNA-seq of 7 million cells profiled, 3 million failed, with only 4 million high-quality cells left for the clustering analysis, as well as a whole brain, a Murphy data set with 4 million cells segmented as well. So the combination of the RNA-seq and the Murphy data together allowed us to define this higher resolution cell type atlas with a unified clustering approach that generated more than 5,300 individual clusters. And we organized the 5,300 clusters into yet another hierarchical structure with higher level descriptions at supertype, subclass, and class levels, as shown in those UMAPs. The UMAPs only show definition at subclass level, and more details cannot be revealed in this whole brain UMAP yet. So one major finding that just directly coming out of this is the extraordinary diversity of neurons across the entire brain. So neurons form 5,200 out of the 5,300 clusters, and also vast majority of the subclasses shown here. A second major finding from this is that the cell types, or these transcriptomically defined cell types, are not only defined by their molecular identities, for example, neurons versus glia or immune cells, or what neurotransmitter each neuron type expresses, glutamatergic, GABAergic, dopaminergic, serotonergic, and things like that. But cell types are very much also defined by the spatial information, the regions where they come from, as you can see in this UMAP colored by the region. So in fact, the cell types across the brain are very much defined by the combination of molecular identity and spatial specificity. So here, I just want to dive a little bit deeper to look into the diversity of the neurons in the brain. So for all the 5,200 neuronal clusters, we grouped them into we separated them into 6 neighborhoods that are based on major brain, large brain structures. Here, this slide shows the 3 neighborhoods So this is all the glutamatergic side-free neuron types that are found in telencephalon, or also called pallium, the pallium structure. And here shows all the GABAergic neuron types found in the subpallium part of the telencephalon. And here, this one shows all the neuronal types from hypothalamus and its closely related extended amygdala areas. In each case, even we dive a little bit deeper into this, we still find extraordinary diversity transcriptomically. But at the same time, with this Murphy's data, it shows that every cell type also has a highly specific spatial location in certain parts of the brain. So, and the transcriptomic variation really correspond well with the spatial specificity and the relationship across those regions. Similarly, the other three neighborhoods, the thalamus, habenula, midbrain, hindbrain, glutamatergic, and midbrain, hindbrain, GABAergic. In each case, there's extraordinary complexity, but also exquisite correspondence between transcriptomic clusters and its spatial location. I also want to quickly say that just from those UMaps, without getting into the details, the UMaps also reveal another dichotomy that we discovered, and that's the dichotomy between the dorsal part of the brain and the ventral part of the brain. Ventral part of the brain, mainly midbrain, hindbrain, as well as hypothalamus regions. In those regions, we find numerous clusters, thousands of clusters in each case. And the clusters are all very small. And the clusters are also, they are discrete, but they are transcriptomically similar to each other, meaning that they have fewer differentially expressed genes between different clusters. So that's the characteristic of the ventral brain. The dorsal part of the brain, on the other hand, the clusters are small, they're also highly located in the ventral part of the brain. The dorsal part of the brain, including cortex, thalamus, and the cerebellum, on the other hand, they also often have large clusters, with each cluster spanning large areas in those brain structures, and gradients. There are spatial gradients, even within clusters across those large areas. So we think this dichotomy between the dorsal brain and the ventral brain transcriptomically may be related to the evolutionary origins of those regions and their evolutionary functions, with the ventral brain mainly carrying out survival function for the organism, including feeding, reproduction, metabolism, the regulation of the basic physiology of the brain. And as such, they are more ancient structures, and also they are under evolutionary constraint. Their cell types have not changed much, and they have this, very peculiar small cluster, very well conserved organization. The dorsal brain, on the other hand, is actually mainly responsible for adaptive function of the organism, sensory motor specialization, cognition, all this newer function that's evolved later in evolution, but also has evolved very fast that allows the animals to adapt to their, particular specific environment. And as such, the cell types are also like that. They rapidly expand into multiple regions, but they are not well, and their transcriptomic profiles also diverge rapidly. But the relationship is not fully resolved yet. So we have all these different gradient, continuous gradient distribution of cell types across the regions. So this is just yet another example, insight that we could learn even from the adult stage transcriptomic profiles to kind of understand where things come from and why, try to explain why the data is like that. Okay, so here I want to use one specific example to illustrate, the complexity that we learn. And this example is to compare the spatial distributions of the different medium spiny neurons in the striatum. There are two major medium spiny neurons, dopamine D1 and dopamine D2, expressing direct pathway versus indirect pathway, well-known segregation of function. But here, and in the striatum, large volume of striatum, dorsal striatum and ventral striatum, we find many clusters, I think altogether 45 clusters as shown in this UMAP. And the clusters are somehow, even in this UMAP, you see that D1 and D2 are well segregated, but at the same time, they're also intertwined. The clusters are lined up, intertwined in this transcriptomic space. And why is that? Why so many clusters? So when you turn into the Murphy data, you figure out why. Each of those individual clusters within those two major subclasses has a highly specific spatial location within the dorsal stratum and ventral stratum where nucleus combines, and also olfactory tubercle at the bottom. And the clusters of the D1 and D2 subclasses correspond with each other spatially, beautifully, as shown here. For every kind of a cluster type, you find a counterpart, in D1 or D2, and this exquisite correspondence of spatial specificity of the individual clusters that are actually reflected in the gene expression gradients we observed. I'm not showing the gene gradients here, but we observed a dorsolateral to ventral medial major axis of gradients that are defined by genes that are also line up all the individual transcriptomic clusters along this gradient, and then with increased spatial specificity. So, and we know that, the people who study the MSNs and the striatum know that there are all these different spatial domains, functional domains that are organized in a very similar spatial pattern. So now we have a transcriptomic map, blueprint for the potential functional specificity emerging out of those structures. Okay. So I talked about non-neuronal cells having not so much diversity as neuronal cells and neuronal types, but they still exhibit a very interesting diversity, especially in the astrocytes and ependymal cell populations. And here I use the corresponding UMAP and Murphy data to show the spatial specificity of astrocytes. There are three major groups of astrocytes. The main group of astrocytes is located within a gray matter intermingled with neurons. And those astrocytes also have regional specificity, lots of clusters corresponding to specific astrocytes in specific brain regions. The second group of astrocytes are called interlaminar astrocyte, or actually recently I just saw a paper describing they are actually the glia limitans superficialis astrocytes, which is a special kind of the GLS astrocytes that line the peel surface of the brain, in both telencephalon and non-telencephalic regions, line the peel surface as well as the inside borders between different brain regions, and they form another interesting astrocytic barrier, between a brain and the ventricular, the peripheral system, or the border of the brain. And then the third major type of astrocytes are those lining the rostral migratory stream. And the rostral migratory stream goes from the dorsal subventricular zone of the brain, where newly formed adult neurogenesis happening, generating olfactory bulb granule cells. And the newly generated neurons migrate into olfactory bulb through the rostral migratory stream. And there's this special group of astrocytes lining the stream, forming the tunnel, allowing the neurons migrate. Some of the astrocytes also have neurogenic potential. And here, the transcriptomic clusters, again, perfectly corresponding to the migration patterns of the cells from the subventricular zone all the way to the olfactory bulb. Here, next slide shows the diversity of the ependymal cells across the entire brain, with, again, many different ependymal cell subclasses and clusters. And they are distributed across the entire brain. They are lining the ventricles, all the ventricles in the brain, medial, lateral, anterior, posterior. They also form parts of the circumventricular organs. All the circumventricular organs that you can find, such as subfornical organ, median eminence, area postrema, things like that. That has very specific ependymal cell clusters. And those ventricles and circumventricular organs are important, again, for brain brain-blood interaction or brain-body interaction in terms of nutrient exchange, hormone exchange, and sensing of metabolites and things like that. And here I just wanted to highlight special ependymal cells that are found in the ventral third ventricle within the hypothalamus, especially the tanycytes, a series of tanycytes lining the ventral third ventricle. And the specific tanycyte subtypes that were well characterized before correspond also specifically to the transcriptomic clusters that we just identified in a completely de novo manner. And I will come back to tanycytes later in my aging study. Okay, so overall, we also identified a large number of gene modules, especially transcription factor co-expression modules that collectively forms a transcription factor code across all the different cell types and across the entire brain to provide this organizing, organizing factor of the cell types in different parts of the brain and define the relationships and potentially their developmental origins as well, as I will also come back later. So finally, for this part of the talk, for this part of the talk, I just want to say that we now present all the single cell RNA-seq data and the morphage data across the entire mouse brain in this Allen Brain Cell Atlas or ABC Atlas web portal to allow people to, users, any of you, to navigate, explore the different cell types, where they are, understand their relationships, as well as all the genes expressed across the entire brain, in all those cell types. Furthermore, we also developed a computational tool called Map My Cells, which is a cloud-based tool that allows users to upload their own transcriptomic or spatial data, any kind of cell by gene matrix, and then map each individual cell of their own data into our whole mouse brain reference atlas to obtain a cell type identity, cluster identity of their cell, of your cells, and to compare with your own clustering result, and also to get the spatial information associated with those clusters, into your own data. And I just wanted to say that again, in our ongoing work, we are also now integrating our whole brain single cell RNA-seq data with a separate set of single nucleus RNA-seq data generated in Evan McCoskell's lab within the consortium. We're integrating them all together to create a more refined whole brain map that includes both single cell and single nucleus. We're also going to be integrating other spatial data from the consortium, such as those from Xiaowei Zhuang's lab. And we are now developing new computational approaches for better integration. And we are developing next generation of Map My Cells tools using the new integrated method, especially based on SCVI-based method. So those will be kind of new versions of Atlas useful to people. Okay, so with that, then, summarize quickly for this part. Distinct features of transcriptomic cell types in different brain regions. We find this kind of integrated molecular anatomical organization. And this is an inference, but we strongly believe that relatedness between the adult stage transcriptomic types actually likely reflects their developmental origins and or evolutionary homology. So you can kind of peek a little bit into the history of those cells, even just from their adult stage transcriptomic data. And it makes sense, in many ways. And I will show you one developmental study to illustrate that. Transcription factors are major determinants in both adult and developmental stages. Okay, so now I'll switch gear to talk about the second study, which is to exactly use the cell type information that we have obtained to understand how cell types change in the aging brain. So we are creating an atlas for a healthy aging mouse brain, and very much driven by the rich knowledge that is already present in the aging field, where many studies, not only in mouse, but not only in brain, but also in the entire body, about the major hallmarks of aging summarized in this very nice review here. There are known major factors that contribute to aging, including, for example, genomic instability, epigenomic changes, metabolism and nutrient sensing, dysregulated mitochondrial function, stem cell depletion, all these different factors that are happening. And we wanted to hopefully use our single cell type specific transcriptomic profiling to see exactly which cell types exhibit this kind of changes. So here in this study, we profiled 16 broad brain regions as shown here, and that cover about 1/3 of the entire brain. And regions include the forebrain, midbrain, hindbrain. And 2 ages of mice, young adult, which is the same as our whole brain data set, and aged 18 months old mice, which are equivalent, people told me, to like 60-year-old people. That's not me. Okay. Okay, so okay. We used the map-by-cell, label transfer approach to assign.
Sorry.
That's okay.
There we go. Yes.
It is English. We used the label transfer to assign classes, subclasses, and supertype labels from the whole mouse brain taxonomy. We also performed a de novo clustering of this data set to identify potentially age-specific clusters. So overall, this is what we got, about 850 clusters. And we used stringent computational approaches, 3 different approaches to identify age-associated differentially expressed genes. Want to make sure that they are well validated in the data. And from this, we came up with about 2,400 HDE genes that are spread out across all these different cell types in different parts of brain, and 14 age-biased clusters. So as you can see here, only 14 clusters that are changed with age out of 850. That means that a vast majority of the transcriptomic clusters are actually quite stable across the different ages. And then there's the major changes in the aging brain actually are reflected at the individual gene level, with those 2,400 genes spread out among the commonly shared clusters. And here are the distribution of those 2,400 HDE genes in non-neuronal types and in neuronal types. We find actually a large number of those HDE genes are concentrated in non-neuronal populations, showing that even though non-neuronal cells don't have as much diversity, they actually are very subject to change. They are very sensitive. It's a lot of changes in terms of the gene expression under with age. So on the neuronal side, so that's why, for non-neuronal cells, we go down to supertype level, whereas the neuronal side, we summarize at a subclass level. And for neurons, we find that there are a large number of gene expression changes relatively in cortex and in hypothalamus. So here shows the correspondence of the HDE genes with different computational method. And here shows that out of those 2,400 HDE genes, actually a very small number of them, about a dozen or so, are shared among many different neuronal subclasses or types in the brain. So those are the universal genes that are changed in many parts and many types of cells. But a vast majority of the HDE genes are actually only found in 1 or 2 specific cell subclasses. So most of the changes, suggesting that most of the changes are cell type specific. And here summarizes the changes in non-neuronal and immature neuronal populations. Number of DE genes shown here, and also the clusters, the age depleted or age enriched clusters. All 14 of them are found actually in non-neuronal or immature neuronal populations. Again, supporting the notion that they are really actually the hotspots of aging. And here, I just wanted to specifically show the 3 major adult neurogenesis niches in the brain. And this first one is the subventricular zone and on top of dorsal striatum that I mentioned earlier, that generate olfactory bulb granule cells and migrate through the rostral migratory stream. The second is in dentate gyrus, the well-known dentate gyrus granule cells that are many of them generated through adult neurogenesis. And the third one is actually the third ventricle, the ventral third ventricle within the hypothalamus, the tanycytes that I told you about. Like the astrocytes, some of the astrocytes, the tanycytes also have neurogenic potential. So we find that in all 3 adult neurogenesis niches, there's the depletion of immature neurons, as well as changes in the associated astrocytes and the ependymal and tanycyte cells. So suggesting a dramatic depletion of adult neurogenesis. Let me show here. And then we also looked at oligodendrocytes in this slide. We see that oligodendrocytes, in the brain undergo dramatic changes. Very large number of DE genes associated with mature oligodendrocytes, as well as many of the clusters that are changed. So what are those? So this is a UMAP of the oligodendrocytes, major types, oligodendrocyte precursor cells, OPCs, and all the mature oligodendrocytes. There are so many of them. And then there are also the intermediate, immature oligodendrocytes, COP, NFOL, MFOL, the intermediate ones. So what we found that actually all the immature intermediate oligodendrocyte populations are depleted in the old aged brain. So they are all like this greenish color, adult enriched. So suggesting a reduction or elimination, diminution of the forming of new oligodendrocytes in the aged brain. We also observe, of course, a lot of gene expression changes, and many of those gene expression changes in the mature oligodendrocytes are related to, myelination, metabolism, lipid metabolism, motility, all these different kinds of gene families, suggesting a decrease of the myelination function, as well as specific clusters, age-associated clusters, especially in the hindbrain, brainstem areas that's associated with many axon projections, a lot of myelination, and that's also under this kind of dramatic age changes. Okay, we then also observe widespread increased immune response and inflammation. And this is reflected, of course, a lot of changes, not surprisingly, in the innate immune cells in the brain, microglia and border-associated macrophages, BAMs. You can see all these gold terms here associated with large number of HD genes. They are all related to immune response, inflammation, cytokine production, NF-kappa B signaling, MHC molecule activation, and also interferon response. So this is the general large-scale gene expression changes within the entire microglia and BAM populations as shown in this UMAP. We also observed the 2 specific age-enriched microglia clusters, 842 and 843, that kind of begin to look like disease-associated microglia with, again, very, very pronounced activation of the immune program. And furthermore, beyond the immune cells, we see widespread increase in antigen presentation involving both MHC class 1 and MHC class 2 molecules in many cell types, not only microglia and BAM, but also vascular cells such as endothelial cells, arachnoid border cells, and also in ependymal cells. Ependymal cells and tanycytes, and even further in some specific neuronal types. These are DMH, dorsal medial hypothalamic nucleus specific neuronal type. Okay, so now we zoom in into the third ventricle of the hypothalamus. Tenocytes and ependymal cells, as I mentioned, they undergo dramatic changes. This showing a lot of gene expression changes that are confirmed by spatial data. And here shows the morphology of the tenocytes. Tenocytes have tantalizing morphologies, with this long processes extending from the ventricle into the nearby hypothalamic neuronal cell space. And people believe that those long processes are important for, information exchange, nutrient exchange, hormone exchange, interacting with the neurons and things like that. So a lot of immune activation in ependymal cells, decreased neurogenesis, particularly pronounced in tenocytes. We also even find increased number of dams within the ventral third ventricle areas, especially in the median eminence areas, so that suggesting dramatic changes. And then furthermore, then we look into the neurons in this region, hypothalamic neurons, because we find that in all the neuronal populations, we find a particular group of neuronal cell types, subclasses, that have larger number of HDEG changes that are all highlighted here. Some of them are cortical neuron types. One of them is in brainstem, but most of them are in hypothalamus. So all the red highlighted neuron types with big HD genes are found in hypothalamus. And where are those neuron types? We find that the vast majority of them, again, are concentrated right next to the third ventricle, the ventral cervical. They are located in regions such as arcuate nucleus, dorsal medial nucleus, and the paraventricular nucleus along the third ventricle. And then what those cell types are. So now we zoom even deeper, not only at the subclass level as shown by those labels, but also the supertypes and clusters within those subclasses. We find highly specific neuronal clusters that have large number of HDE genes, and some of which also have upregulation of MHC class 1 molecules, which I don't understand why yet, and we're very intrigued by this. And those specific neuronal clusters are also specifically associated with neuropeptide genes that are known to be involved, and their receptors that are known to be involved in food intake and energy homeostasis. So this includes this particular cluster, transcriptomic cluster in the arcuate nucleus that express AGRP and MPY, the known hunger peptides in the brain, and their receptor for this other arcuate nucleus cluster. It also includes paraventricular nucleus clusters that express CRH, a stress hormone, and also expressing vasopressin and oxytocin, which also play a role actually in energy homeostasis. And finally, I wanted to point out another cluster. This is the DMH specific cluster, which I showed you earlier. This one has a lot of gene expression changes and upregulation of MHC1, and it's the one that expresses GLP-1 receptor and leptin receptor. The sensing peripheral peptides that are, again, regulate appetite. So, okay. So that then to summarize the whole aging story, we found 3 major type of changes, dramatic changes in the healthy aging brain. The first is the decrease of the neuronal structure and function, and that's reflected in decreased adult neurogenesis, decreased neuronal signaling, and decreased neuronal structure integrity. That's mainly reflected actually by changes in oligodendrocytes, myelination, and other things that oligodendrocytes do. The second major change is the increased immune response and the inflammation that is also widespread in the aging brain, not only in microglia and BAMs, border-associated macrophages, but also in some of the vascular cell types. And finally, we find this intersection point, a hot spot of aging, early healthy aging, that's concentrated in the ventral third ventricle of the hypothalamus, and involving cell types that are important for regulation of food intake and energy homeostasis, including ependymal cells, tenocytes, and a highly specific set of hypothalamic neuronal types. Okay, so summarizing this part of the talk, aging brain, the decreased neuronal function and increased immune response, and the hypothalamus region appears to be a hotspot for aging, and that involved in food intake, energy homeostasis, and simultaneous decreased neuronal function and increased response. And I just wanted to say that many of the cell types that we discovered in this aging study that exhibit selective vulnerability in healthy aging, many of them are concentrated at the brain-blood interface. All the ventricles, neurogenesis niches, hypothalamus, immune cells, vascular cells, ependymal cells. It shows a strong tie, between brain and body and the interaction, and neuroinflammation, energy homeostasis. So just get us really, really interested in how brain interacts with body and how that interaction actually changes as the early sign of aging, at least in the mouse brain. Okay, so with that, without much time left, so I will quickly go over the third study I want to tell you about, and that's switching into development and back into one brain region, one cortical region. So in this study, we created now a complete developmental cell type atlas for one cortical region, but covered then with dense temporal sampling for the entire prenatal and postnatal developmental process of all the cells within this region. Okay, so here shows the 35 individual time points we have profiled, actually using two data sets, single cell RNA-seq and multi-omic. Okay, so here is the UMAP showing the developmental cell type atlas we built using the single cell RNA-seq data. Again, definition of major cell types, class, subclass, cluster, and even subcluster, as well as how they, progress through age. And I just wanted to highlight one critical age point, and that's the perinatal aging point, before and after the puppy is born. So where is this time point in the UMAP? It's right here. Okay, so anything here are prenatal, and all the things after that are postnatal. Prenatal involves the main neurogenesis process with all the neuronal types generated, major class, subclasses generated. But what's amazing shown here is the tremendous transcriptomic diversification of already all the born neurons in the postnatal developmental process coincide with the circuit formation and maturation. So we can see that there's actually much more transcriptomic changes after the neurogenesis is already completed, suggesting continued diversification. So, okay, so here, just a couple of slides to show that. This is actually a summary of the entire trajectory of prenatal neurogenesis and gliogenesis, showing how the major subclasses of neurons are generated and their time course from the progenitor cells, NEC or radioglia, progenitor cells. And so the conclusion from this part is that prenatal neurogenesis and gliogenesis process is actually a staggered parallel process with the emergence of all the different kinds of excitatory neuronal subclasses and glial cell subclasses. Of course, the GABAergic subclasses come from subcortical regions, as I mentioned earlier, and they migrate into cortex, but they also go through this kind of process in the embryonic prenatal stages. And here shows the complete developmental trajectories that we have computationally derived from our single cell RNA-seq data. Because of the dense temporal sampling we did, we are really just able to obtain the cell type identities from the adult stage and step-by-step propagate them backwards through early developmental stages. purely transcriptomically linkage the cells at different ages together to create this trajectory map. Okay, so this is purely data-driven without involving any lineage tracing study. And we think it's pretty accurate. And the good thing about this is that every time point, the transcriptomic profiles are associated with the cell type identities in real time, right? Because everything's corresponding with each other. The trajectory map is built on top of the transcriptomics data. So without getting into details, this is the glutamatergic trajectory map. Those two are the GABAergic trajectory map for MGE-derived and CG-derived GABAergic neuron types. And this is the non-neuronal trajectory map. So what we found here is that in this entire postnatal stage, there are many more refined neuronal types, beyond, within those subclasses, the clusters that are emerging at different stages of postnatal development, including early postnatal days, eye-opening, which is the mid postnatal days, and the critical period, which is the late postnatal days. So suggesting continuous diversification that's concomitant with the maturation of the circuit. And here, just wanted to quickly show the correspondence between the developmental trajectories and the triple modality, MET types, morphoelectrical transcriptomic types that we defined earlier from our patch-seq data. So this is, and use the parvalbumin and somatostatin GABAergic neurons as example. So this is the developmental map. And here we labeled all the MET types for parvalbumin here, different types, layer-specific ones. This is the chandelier cells. Similarly, for the somatostatin MET types, they have very good correspondence at the cluster level among the different trajectory trees. And we even identified the intermediate MET types that we previously identified from the morphoelectrical studies between parvalbumin and somatin. And we found those intermediate types, one in superficial layer, one in deep layer, map exactly at the junction between the parvalbumin part and the somatostatin part of the developmental trajectory tree, suggesting that their morphoelectrical intermediate property actually is related to developmentally where they come from, right in between the two. So it's good to see those two different data sets corresponding with each other. And here just a slide summarizing as an example, the large number of gene expression changes before and after this eye-opening period. I don't want to get into details, but it's a lot. And finally, in just the last couple of minutes, I want to tell you about the correspondence between the epigenomics data, the ATAC-seq chromatin accessibility data, and the RNA-seq data. So because we now have cell type specificity and age specificity, and we can now investigate the correspondence between all the differentially accessible peaks in ATAC-seq data and all the gene expression, differentially expressed genes in the RNA-seq data across those two dimensions, cell type and age. And we identify all the correspondence, for all the classes of cell types, not only in the excitatory neurons shown here, but also GABAergic and non-neuronal. So we find the corresponding set of peaks and a corresponding set of RNA, the genes that are well aligned, that are now all aligned with each other in cell type specific and temporally specific manner. And then from that alignment, then we can begin to derive a developmental gene regulatory network that is cell type specific by linking things across the triplet, by doing this triplet correspondence. So the triplet starts with the motifs, transcription factor binding site motifs that are found in the specific chromatin accessibility peaks. And because we can identify them, we find associated corresponding transcription factors. We can find specific transcription factor that has the expression matching to that motif from the transcription factor family that correspond to those motifs, the expression pattern. And then at the same time, we also find the peak the peak modules that contain that motifs at different cell types and different developmental stages. So without getting into the details, so here is the gene regulatory network that we find. We are able to link master regulating transcription factors to their downstream target genes in specific cell types and at specific times through the intermediate, which are the motifs, transcription factor motifs that are found in the chromatin accessibility peaks. So here's just another example. We're doing it again for a different group of cell types. And we also can find like how they relate to each other through the shared transcription factors. And finally, I just wanted to show this, that we find complex regulatory landscape, even like for one gene. And here I want to use one gene, the COX-2 gene, as an example. The COX-2 gene is actually a pretty large gene with a gene body covering about 200 kb, multiple exons shown here. And also the COX-2 gene has complex expression patterns across different cell types. It is a well-known upper layer cortical excitatory neuron marker, but it also has differential expression at different developmental times in other cell types. And we find all these different peak modules that are associated with this one gene. And we find that the different peak modules or different specific peaks are associated with the same gene, but in different cell types and at different times. So you can understand that this is one gene that has this specific cell type and temporal pattern, and its expression in each cell type and each time is associated with a different set of peaks. So those peaks are more specific. Collectively, they regulate this dynamics of the same gene in different cell types. Okay, so summarizing this part, we observe continuous cell type diversification throughout the development. The early stage with all cell classes and subclasses defined by neurogenesis and gliogenesis, but the later stage, increasingly refined cell types emerge during postnatal neuronal maturation and circuit assembly, including the late emergence of many new types, clusters in the eye-opening stage and in the later critical period. We find a cooperative dynamic changes between chromatin accessibility and gene expression. And we define this kind of gene regulatory networks. And finally, this point, as I said at the end, a single gene can be regulated by multiple peaks that are associated with different cell types and or different developmental stages. So we're now able to tease out the dynamic changes. With that, I just want to thank a large team of scientists and investigators at Allen who work collaboratively on all these different projects. And in particular, those are the people who have led the whole mouse brain cell atlasing work. And also wanted to thank Kelly Jin, leading the mouse aging study, and Yuan Gao, a scientist, both are scientists leading, Yuan lead the developmental study. Okay. Thanks for your attention.